Hello, my name is Cipriano DeAbreu, and today I would like to show you part of the results of the research that we've been conducting at the Mycoplasma Research Laboratory of the University of Minnesota. The title of my research is the characterization of a Mycoplasma pneumonia experimental aerosol infectious model in pigs. First of all, let's talk about Mycoplasma pneumonia. Mycoplasma pneumonia is a small bacteria that is known for being virtually present in every single country where pigs are produced and raised. Mycoplasma pneumonia is a bacteria responsible for producing a disease in the pigs known as enzootic pneumonia. This disease can be transmitted through different routes: direct contact in between an infected and a non-infected animal, and also through aerosols when an infected animal coughs or sneezes. Mycoplasma pneumonia is known for colonizing the swine respiratory tract, and once it is present in the respiratory tract of the animal, it can impair the mucosal clearance mechanisms, opening a pathway for other bacteria and viruses to colonize the swine respiratory tract. Mycoplasma pneumonia is associated with high economical losses each year in the swine industry. Those losses are estimated to be around $7 per each marketed pig, and are mostly associated to the increased use in the antibiotics to fight against this bacteria, and also due to the increased time needed for the pigs to reach the market weight. What is an experimental model of infection? An experimental model of infection is a collection of different techniques that are used experimentally in order to replicate a particular disease in an experimental subject. In the case of Mycoplasma pneumonia in pigs, different models of infection have been described previously, including the intranasal inoculation, intratracheal inoculation, and the aerosol inoculation of the animals. Why are the aerosols so important? They are believed to mimic better the natural route of transmission of the disease in between an infected animal and a non-infected animal, and by studying them, we would be able to better understand these bacteria. The objective of our study was to establish and characterize an experimental aerosol infectious model for Mycoplasma pneumonia in pigs, and the way how we accomplished this was by exposing 4 different groups of pigs into different combinations of doses and volumes of an infective inoculum containing Mycoplasma pneumonia. Pigs were exposed to aerosols that were produced using a nebulizer that was coupled into an isolator system, where the piglets were placed individually one by one. Once all the piglets in the 4 different experimental groups were exposed to the aerosols containing Mycoplasma pneumonia, they were followed over a period of 28 days. During this time, different samples were collected, and with those samples, we were able to assess the detection of the bacterial DNA in the swine respiratory tract. We were also able to assess the bacterial load in the swine respiratory tract. We were able also to assess the humoral immune response in the swine respiratory tract against these bacteria, and also in the serum. And lastly, we were able to measure clinical signs and lung lesions associated to the bacteria on the animals. By the end of the study, we were able to detect the appropriate combination of doses and volumes that were able to replicate better the disease using aerosols as the route of inoculation. So as an outcome, we were able to detect an effective combination of doses and volumes that were able to replicate experimentally the disease. By characterizing and studying an aerosol infectious model in pigs for Mycoplasma pneumonia, we would be able to test more accurately new vaccines and treatments in order to fight against these bacteria on the field. This also gives us a tool to experimentally study and understand the bacteria, and all this new knowledge that can be generated using aerosol infectious models for Mycoplasma pneumonia can then be applied back into the field in order to improve the animal health and decrease the economical losses associated to the bacteria. Lastly, I would like to acknowledge the 2 companies that funded this research project, and also the Mycoplasma Research Laboratory for their continuous help during the development of this study. Thank you.